Differential centrifugation. So this is in the 3.2.1.3 methods of studying cells. So differential centrifugation is the most common method of fractionating cells, and fractionation is basically the separation of the different organelles within the cell. So the first thing that you would do is cut the tissue in an ice-cold isotonic buffer. So it's ice-cold to stop enzyme reactions happening, and it's isotonic to stop osmosis, so that if the solution is too dilute, so water doesn't move into the organelles by osmosis and burst them open. And then you've got a pH buffer to stop changes in pH, because obviously extremes of pH can denature enzymes. Then what you would do is grind the tissue in a blender or homogenizer. This breaks open the cells, releasing the organelles. Then you would filter to remove insoluble tissue or remove the debris. Then you put the filtrate into a test tube and you put it into this machine, which is called a centrifuge, and it basically just spins around the test tube really, really fast. So first of all, you would centrifuge it at low speed at about 1000 G for 10 minutes. And the first thing to form a pellet, which is like a layer at the bottom of the tube, is the densest organelle, which is called which would be the nuclei. And then you would keep that pellet, take it out of the test tube and keep it. And then the rest of the fluid that's left over is called the supernatant. Then what you would do is spin the supernatant at medium speed for 10,000 G for 30 minutes. And this will pellet out the mitochondria, which are the second densest organelle. Then you would centrifuge it at really high speeds, which is about 100,000 G for 30 minutes. And this pellets out the Golgi apparatus. And then finally, you do it at very, very high speeds at 300,000 G for 3 hours. And this will pellet out the smallest organelles, or the least dense organelles, the ribosomes. So the reason that we do differential centrifugation is because it allows us to look at the function of the organelles within the cell. We can look at the individual organelles and study them in more detail, and this helps us to determine each organelle's function within the cell. So here's the whole process. You'd first of all, say if it was liver or a big piece of tissue, you would cut it up into tiny little pieces in the ice cold buffer solution. So it's got to be ice cold to stop enzyme reactions. It's got to be buffer to keep the pH the same so that the enzymes denature. And it would also be isotonic so that water doesn't burst open the organelles when it moves in by osmosis. Then you would put the chopped tissue into a blender or a homogenizer, which break opens the cells. Then you would filter the mixture to remove the debris. Once you've got the filtrate, you then put the filtrate into test tubes and put it into a centrifuge. And you spin it, first of all, at low speed, and this will form a pellet of sediment, which would be the nuclei, the densest organelle. Then you would spin it at the supernatant, you'd spin that at medium speed, and that will pellet the mitochondria. So then you'd take the mitochondria away, and you would spin the fluid again at high speed, and you would get the Golgi apparatus. And then at very high speeds, you would get the least dense organelle, like the ribosomes. So remember that the homogenizer is to break open the cells to release the organelles. The isotonic solution is so that damage isn't caused to the organelles by osmosis. A buffer to keep pH the same so that the enzymes don't denature because of extremes of pH. It's ice cold to slow the enzyme reactions, and we filter it to remove the debris or insoluble tissue.